So this is actually a continuation of a lecture I gave you before. And as neurobiologists, one of the things that's really incredible, if you think about it, is the fact that when you are looking within our discipline, I gave you the lecture last week, and then immediately after that lecture had come out, where I said that there were no therapies that were currently available for Alzheimer's disease, a paper came out almost immediately after saying exactly the opposite, that there is a lot of potential, there's a therapeutic benefit to be able to deliver what's known as microbubbles and to use a non-invasive technique like ultrasound. And it's a technique that allows us to go back and revisit a lot of the issues that were covered originally in our Alzheimer's disease lecture. And I also want to make sure that you understand some of the rationale behind the paper if you indeed read through that particular paper. So we're going to be looking at this in a number of different ways. One, we're going to look at it just for the technique itself. It's a really cool technique, and if it actually does pay off its dividends, the way that it's going to work is going to revolutionize the way that we do clinical treatments for a number of neurodegenerative diseases. It does not have to, by any means, be isolated only to Alzheimer's disease. It could, in theory, be used for prion diseases or other types of disorders that we've not really covered so much in this particular class. In addition to that, we're also going to revisit Alzheimer's disease and also understand a little bit more behind the paper and why different things are used in this particular paper. You do not have to read it. However, it's a short read in Science Translational Medicine, and it's something that maybe as you go through, you might be interested in taking a look at. So I'm going to talk to you a little bit about scanning ultrasound today, and they're going to abbreviate scanning ultrasound as SUS. And one of the things that we talked about was this deposition of beta amyloid. And this amyloid beta or beta amyloid seems to be one of the initiating events. We're not exactly sure why it does it or how it causes neurodegeneration, but it seems to be one of the precipitating events that occurs in neurodegenerative conditions that underlie different forms of Alzheimer's disease. And in this particular case, they haven't done it clinically yet. They're very close to being able to do it clinically, but they make the proof of concept in an Alzheimer's disease mouse model, and we'll go through that a little bit more. So we're going to go back and revisit Alzheimer's disease just as a cursory overview. It should be a review for you, so if there are things that are missing in your background, it's time to kind of circle those off and maybe think about coming in to visit me to go through some of the lecture material. In addition, I'll start to introduce you to this new technique and microbubbles. If you've never heard of microbubbles in health and disease and neuroscience and neurobiology, it's one of the hottest topics that theoretically has a lot of therapeutic potential. There are individuals here at the University of Toronto, not in departments that you're aware of, but in departments like the Institute of Biomaterials and Biomedical Engineering, who are actually using microbubbles as a therapeutic tool. So it's useful to know a little bit about ultrasound and how they can theoretically clear different types of oligomers related to neurodegeneration. We'll be talking about different transgenic mouse models. When you're reading through a paper, one of the things that you should realize is there are a number of transgenic mouse models. And I had mentioned that there are two broad categories. One that is related to amyloid precursor protein. The second that is related to tau and tauopathy. In this particular case, they decided to use the amyloid precursor protein transgenic animal. But even in that grouping, if you go back to the slides, there are probably a good 7 or 8 different models, transgenic models that involve APP. Why did they choose to use this one here? And when you're reading through these papers, you should always be careful and understand why they're doing the types of experiments that they are. I'm going to go through a couple of technical details that I would like you to make sure that you are familiar with and understand, because as neurobiologists, it's not about getting a cursory overview of neuroscience. It's to actually start thinking in your 3rd and 4th years about how impactful and why experiments are done in the way that they are. So going back and looking at Alzheimer's disease, again, the major precipitating factor seems to either involve hyperphosphorylation of tau and or this beta amyloid. Most individuals now will be firmly entrenched in the camp that says that it's the amyloid beta cascade, the initiation of beta amyloid outside of the cell, that actually begins this entire process as they start to aggregate into these larger extracellular fibrillar deposits. As Dr. Kalia mentioned to you before, it is very, very likely that prior to the formation of these large extracellular fibrils, that one of the things that's been happening is that there has been an aberrant accumulation of smaller molecules. And she had talked about that. She had talked about the idea of seeding and potentially these things looking a lot like prions. And so it's not just the same, it's not just Alzheimer's disease where you have misfolded proteins like beta amyloid. It might be similar to alpha-synuclein that Dr. Kalia talked to you about on Thursday. So again, beta amyloid seems to be elevated within the Alzheimer's disease brain, either because we are producing too much of it, and we know that there are genetic factors that lead to the overproduction of different beta amyloid precursor molecules, and you should definitely go back and make sure that you review that, or it might be due to its impaired removal. And we talked about microglial cells and ApoE as sort of the clearance related to these beta amyloid deposits. So all of this is background for you. All of this should sort of ring a bell in terms of the mechanisms that we've looked at previously. So I told you at the very end of the last lecture that I gave you on Alzheimer's disease that there are a number of different therapeutic approaches that have been taken. And these are actually outlined for you in this particular paper in one paragraph at the very beginning in the introduction. And a lot of the therapeutic strategies, as I mentioned to you, have involved either inhibiting those different secretases that are involved in the formation of these different amyloid beta particles, or into the increased removal or clearance of these as well. And some of that might be mediated through the different types of antibodies that might be delivered in terms of passive immunization approaches or active antibody delivery. All of those approaches to date have not been very, very successful, not even in animal models. And there seems to be a lot of confusion as to whether or not passive immunity, active immunity is the better approach in terms of clearing off using antibodies this beta amyloid overproduction. And again, one of the biggest issues pharmacologically in inhibiting these different secretases, like the gamma secretase that we mentioned in the last lecture in Alzheimer's disease, is that when you're inhibiting this, you're also inhibiting gamma secretases outside of the complexes that form the amyloid beta. And there are many off-target effects. So you will not just affect the pathway that forms the different beta amyloid peptides, but in addition to that, you will have many, many other substrates being affected that may be deleterious to other processes going on in the body as well. The last thing to make note of, when you're hearing about this in your fourth year courses, whether it's in LMP or in other courses, this passive immunization process is actually very expensive. It's not something that's trivial. The production of different types of antibodies that are used for delivery, even through recombinant techniques, is orders of magnitude much more expensive than the technique that I'll be showing you here today. And if you take into account all of the statistics that you've been hearing in different conferences, workshops, in this class and elsewhere on the news, that you'll know that if the average person, it costs about $500 to do this passive immunization treatment, and those numbers are going to go up to tens of millions of individuals, it's just going to overwhelm our healthcare system. We won't be able to actually provide passive immunization of antibodies to prevent the onset of Alzheimer's disease if it continues on in the cost ineffective manner that it currently is. So people are looking for alternate therapies for a number of reasons: off-target issues, cost issues, as well as ineffectiveness overall. So let me tell you a little bit about how this ultrasound technique works. It's been used in a number of different situations in a number of different ways. If you are not aware of what microbubbles are, by definition, they are micro, and by micro, we mean they're on the order of microns. In this particular paper, they meet the criteria exactly in terms of being less than 10 microns. The microbubbles that they are generating and that they go back and check for size are on the order of 4 to 5 microns in diameter. And these microbubbles are exactly what they sound like. So these are biologically inert, and these are either a lipid outer coating or a polymer outer coating that usually involves peptides. And then there's a stabilized gas core that surrounds it. And so one of the things that happens when they get excited by different sound waves like ultrasound, they will start to expand and contract. And these microbubbles expand and contract. And as they do so, they have the potential to go and disrupt the blood-brain barrier. So these microbubbles that I just mentioned to you that are on the order about 4 to 5 microns or less than 10 microns, depending on your target tissue, are injected systemically. They do not have any deleterious effects outside of being injected. And again, in this particular paper, they used a lipid shell. You could use a peptide shell or whatever you want. And these microbubbles expand and contract when there is acoustic pressure applied to them. Again, ultrasound is acoustic pressure, and the rarefaction as well as the compression, the sound waves, in other words, the compression and expansion or rarefaction over several cycles leads to them being acoustically activated. They will also expand and contract. And when they undergo this acoustic activation, one of the things that happens is that they will start to displace the vessel cell wall. In other words, as they expand and contract and as they brush up against the blood vessels that they're flowing through as you're providing focused ultrasound on an area of the body, one of the things that happens is that they will transiently open tight junctions which are associated with the blood-brain barrier. And so by doing so, you have now opened up the blood-brain barrier. And again, all it requires is the opening of this blood-brain barrier and the minimization of the tight junctions that are found in this blood-brain barrier by injecting an inert substance and then just using ultrasound. Is that clear for everyone? Have any of you actually heard of microbubbles and ultrasound techniques? Some of you have? Okay, great. So if you have any questions related to this, I'll try my best to answer them, and you might want to jot a couple of questions down as we're going through. So this gets into this whole issue. So there's an issue related to the ineffectiveness of a lot of the different strategies, and I mentioned those at the end of the last lecture. Off target, if you simply inhibit gamma secretase, which sounds in theory like a great idea, or you cause another secretase, the non-amyloid pathway, to become activated, again, it sounds like a great idea, but there are the potential to produce a lot of other substrates that might be deleterious for your body. And in fact, many animal models have suggested exactly that. There are cost issues, and so people have been looking at alternatives for this. And again, in terms of the safety and efficacy of this, there has been other studies. And one of the studies that have actually looked at the blood-brain barrier, not in a mouse model, but in a model that involves using primates, that if you inject microbubbles, slightly different, but overall the same idea, and you use ultrasound in an area like the visual cortex, just to see if it causes any damage to that area of the brain, by using this focused ultrasound, the monkeys were still able to perform visual tasks. Again, suggesting that the introduction of these microbubbles and ultrasound does not cause any permanent damage to any of the neurons or any other structures in the visual cortex that was involved in visual spatial tasks. And these complex visual tasks were not affected at all, suggesting very strongly that this is a safe and efficacious technique. And again, these are not the same authors that performed this. Again, if you're really interested in it, there is a link in the paper that I showed you at the very beginning that was published in Science Translational Medicine that will actually take you to this paper that was published 2 years ago. So one of the first things that these authors decided to do was to actually do what's known as a proof of concept. So can they actually show and demonstrate that using just microbubbles, so you're injecting in these polymer shells, lipids, filled with an inert gas, injected into an area of the brain, and then use this particular device that is known as TIPS, or Therapy Imaging Probe System, and to either do focused ultrasound, just like in that previous paper that I just showed you, focused ultrasound on the visual cortex, or could they actually run it across a big area of the brain? Now, again, there's a couple of things in this proof of concept that as neurobiologists, you should all be sort of aware of. One is that they injected a dye systemically known as Evans blue. So those of you who are in physiology, tell me a little bit about Evans blue. What does it do? What is it used for? putting the physiologist on the spot. Does anyone know? Evans blue is a blue dye, as the name suggests. Does it cross the blood-brain barrier? Yes or no? No. Why do you say no? Yes. Sure, but it's a dye. Like if I inject a dye, don't you expect it to cross the blood-brain barrier? It's not a charged dye. So in theory, it should cross. Why doesn't it cross? It doesn't cross, by the way, and you should know that. Evans blue by itself does not cross the blood-brain barrier normally unless it's disrupted. But how or why does it not cross the blood-brain barrier? It's a simple dye. It's a non-charged dye that you're injecting in, and it's been used in physiology for decades. Evans blue does not cross because it binds to proteins. What's the most predominant protein that you have in your bloodstream? Albumin, right. So albumin does not cross the blood-brain barrier. This dye that binds to albumin also does not cross the blood-brain barrier. Perfect. So you need to know that Evans blue binds to albumin. It's important because I'll come back to it at the very end of today's lecture as well. So it binds, it doesn't cross the blood-brain barrier. They have injected in this animal here Evans blue dye. They have not used ultrasound, and you do not see any blue in its brain at all. Here in this mouse, they have injected microbubbles, and they've injected Evans blue dye. And when they do focused ultrasound, I hope that it's clear, at least on your slides, if it's not clear up here, that you'll see that right here in the area where they did focused ultrasound, that you can actually see visibly the presence of the Evans blue dye, suggesting that the blood-brain barrier does indeed break down using these microbubbles and a marker like Evans blue. And this is just focused ultrasound that is being shown to you in B. And again, it's a proof of concept. Can we get a blood-brain impermeable dye across using microbubbles and having this focused ultrasound? Yes, you can. So can we do scanning ultrasound? This is just a small area of the brain that is opened up. Can we do exactly the same experiment? Inject in Evans blue dye, and instead of using just visible sort of light, can we actually use a system that is shown to you here, known as the Odyssey fluorescence LICOR system? You don't need to memorize the name. You can actually take a dye and have it fluoresce using different types of microscopy. And when you do so, can you see how much dye is actually getting in? So they've done these same experiments here, but instead of using it on a focused area, they have injected in Evans blue dye, and they have scanned across multiple areas of the brain. Not just one area, they've opened up many parts of the blood-brain barrier. And when they go back and take a look at where Evans blue dye is in serial sections, so this is the olfactory bulb at the front, going all the way through rostral to caudal, when they do, they see the dye has spread everywhere. In other words, this is very efficacious. Using scanning ultrasound, they can actually open up the blood-brain barrier. This dye goes everywhere in the brain at different levels. So this is less and this is more. And you can see that the dye gets into many, many different areas in different concentrations. Is that clear for everyone? Yes? Good. So even 1 hour after this scanning ultrasound was performed, you can actually see widespread widespread Evans blue staining in the brain of these different animals. And again, on basic histology, when you go back in and take a look, this is the sham animal. And one of the things that you see, they've been injected with Evans dye blue. They have also been injected with microbubbles. They have not had any ultrasound applied to them. Their brain morphology looks perfect. And when you go back in and do everything exactly the same, inject in Evans dye blue and microbubbles, and you now do scanning ultrasound, you'll see that the dye visibly is spread throughout the entire brain. And I'm hoping that's obvious for you. Yes? Okay, perfect. So this slide and the slide that accompanies it are actually the same slide. I didn't want to fit everything on here for you. So one of the things that they wanted to make sure was not happening was, is the ultrasound, the scanning ultrasound, is it actually causing neurons to die? That would be a bad thing. If you're ever going to use it in therapy in the human brain, if ultrasound, because it's the compressed sound waves and the expansion of sound waves, it would be an incredibly bad thing if all of this worked because neurons are dying and because it's actually causing cell death. And so using a number of different standard histological and histopathological techniques like hematoxylin and eosin staining, they saw no differences either in the sham or in the experimental animals of any changes in structure, nor any forms of widespread cell death. And one of the things that they mention in the article, when you go back and you do a different stain, this is H&E, hematoxylin and eosin stain, and you can see the dentate gyrus and the hippocampus. It looks beautiful. It is not damaged in any way after scanning or after injection. And if you were to take a higher power magnification of it, if neurons are dying and you do a different stain known as a Nissl stain, one of the things that happens is that you would see these little elements that are appearing here on the right of your screen. And these little fragments that you see are actually cells that are dying, and these are known as dark neurons. And on a Nissl stain, which is a different type of stain here, you will be able to see if cells are actually dying. They did not observe that in any of the sections that they looked at. And this is just to show you that this is what a normal section would look like, where you see nice nucleoli, the little dot in the center of the nucleus, as well as a nicely smooth stained cytoplasm, and you don't see any of these dark neuronal bodies. Again, one of the other things that you can do that is often very important. So you've opened up the blood-brain barrier. What do you expect might happen? What might happen if you open up your blood-brain barrier? Yes. You might get toxins. What else might theoretically occur? Yes? You could. And so what might happen if you either have more toxins or if you have disrupted the ionic balance? What might you expect? Sorry? You might expect cell death. So they've ruled out cell death is occurring. What else might you expect? Sorry? Yeah, you might expect different things like inflammation. The other thing that you might expect is that you might actually have a gradient that might lead to ischemic damage. And when you're looking for ischemia as another histological marker in the brain, and you might hear a little bit about it next week when we learn about stroke, one of the stains that you can do is acid fusion. And if you have an acid fusion stain, it's typically used to demonstrate that stroke or inflammation is occurring in the brain. None of these have occurred at all. And again, if you take a look, this is a beautiful stain in H&E. Do you see any blood anywhere? So has there been any leakage of blood out of the blood vessels? So if you did, you would see like lots of little blood vessels here within the like the blood vessels up here or in this section. You might start to see blood in some of the different ventricles, and you don't see any blood in any of the regions that you don't expect to see them in. So this actually tells you what was going on in that previous slide. So this procedure, this scanning ultrasonography actually did not cause any of these dark neurons. There was no cell death as revealed by Nissl staining that the authors went back and looked at. There was no swelling, no infiltration, no changes in ionic balance that they could see that was also causing erythrocytes to move out of the blood vessels into different areas by H&E staining. And again, acid fusion staining also showed no evidence of any ischemic damage. So in other words, this technique, although it allowed the dye to move out and through the blood-brain barrier, it did not cause the the blood vessels to become leaky, did not cause inflammation, did not cause ischemic damage, it did not cause cell death. In other words, it was safe, at least in this mouse model. Is that okay for everyone? Okay, so one of the things that they wanted to do was to do multiple rounds of this scanning ultrasonography. So they do this more than once. They also wanted to, in their transgenic animals that I'll show you in a moment, these APP23 animals that they're using, they wanted to know is at the end of this, because these are the animals that will eventually have an Alzheimer's-like phenotype that are related to memory. At the end of this, can we actually go back and see if there is any benefit to doing any of this ultrasound in this model for Alzheimer's disease? And this is sort of their experimental model. They do behavioral testing, they do histology, as we've already seen, they do some protein chemistry, and they do what's known as an immunoblot or enzyme-linked immunosorbent assay, or an ELISA. And we'll see all of these in a bit. So this, again, treatment scheme was really important. And I'm not putting this up here for you to memorize, but it's more for you to understand. In their materials and methods, they take great pains in telling you a couple of different things. First, the type of mice that they're using. These are APP23 mice. It means nothing to you right now, but it should by the end of today's lecture. The median age at which they started these experimental protocols is at 12.8 months. Again, keep this in the back of your mind. You'll need this in about 4 or 5 slides. The other thing is that they were using male mice, okay? Sometimes you'll see that and it's a throwaway, and some of you will have heard that, yeah, usually in science we use male mice. Here, there's actually a specific reason why they're using male mice. It has nothing to do with hormones, by the way, and you'll see why that's important in a moment. And again, these mice either receive ultrasound or they receive sham, where they're put in. Everything is done the same. They just don't turn on the ultrasound. And this goes on for about 6 weeks, and at the end of the six weeks, they will do a behavioral test in the Y maze, and they will also have their brains taken for Western blotting, ELISAs, as well as for microscopy. I'm hoping this works. So let me go through a couple of things with you here. I told you that they use the APP23 mice. You will hear in the literature about many different types of transgenic mice. These were first originated in 1995. As soon as molecular techniques were sophisticated enough to create these transgenics, and enough was known about the amyloid cascade, people started going out and creating these different transgenic mice. And this first transgenic mouse model that was created in 1995 actually used a PDGF promoter, or that platelet-derived growth factor promoter, that expressed the human amyloid precursor protein. And you ended up expressing these different fragments, the amyloid beta 1 to 40 or the 1 to 42. And as you would expect, because you're overexpressing the amyloid, you have 5 to 14 times higher levels of the toxic fragments that we talked about later. They display many of the things that we've talked about. They have synaptic loss. Synapses are gone in this mouse model. They have the reduced size in the hippocampus. The fornix is also misshapen. The corpus callosum gets affected in this mouse model as well. And they also have memory loss comparable to that found in human AD patients. So if this is such a great model, why don't we use this as much in the literature as the other models I'll be showing you in a moment? One of the biggest issues is the deficits that you see in memory, cognition, etc. in these mice, and it definitely is shown in older mice, correlate with increasing levels of amyloid beta, exactly as you'd expect. The more amyloid beta you have, the more cognitive deficits that you would expect, especially within the hippocampus. However, the same cognitive deficits were also found in younger animals, which had nothing to do with the amyloid burden or the amyloid load. So in other words, if it's only due to amyloid in this model, and that's the only thing that we're overexpressing, why is it that there is no gradation where memory gets worse and worse with age, instead of having younger animals also being affected, even before they have these amyloid deposits or any changes in the hippocampus? And again, as scientists, you would say, well, yeah, it doesn't make really good sense. So this model doesn't tend to support what is now known as the amyloid cascade hypothesis, and it's not the best model. There are a number of reasons for why this model might actually fail. And one of the reasons is that in the background that these mice are generated against, some people have used the C57BL6J background, some people have actually used the DBA2 mice background, and some have used the Swiss Webster mice background. So you have different genetic backgrounds on which you have this transgenic being overexpressed. And so they're different for every study that people are producing. And because of the lack of consistency, people have moved away from this model, and they moved from the PDAPP model, the platelet-derived promoter model, to another model which was known as the AP P695 model. It's also known in the literature as this transgenic 2576 model as well. Very similar in the sense that you overexpress some of the toxic fragments like A beta 1 to 40, and we talked about this accumulating as soluble and then insoluble fibrils. Same with the A beta 1 to 42 in many of the areas that you would expect. In Alzheimer's, this is where you find those deposits in the frontal lobe, temporal lobe, entorhinal cortices, hippocampus, exactly as you'd expect. However, one of the things that's wrong with this mouse is that you don't really have significant synaptic loss. You get the accumulation of these toxic fragments, but you don't really have synaptic loss, and you don't really have any reductions that are visible in terms of loss of hippocampal neurons. So this, again, is not a perfect model mouse either, and this only came out 2 years after the original PD APP mouse. So there is a lot of literature suggesting that these beta and gamma secretase enzymes that I mentioned to you are really important in terms of the pathogenesis of Alzheimer's disease. And the first mouse model to actually go back and look at the effect of these different gamma secretase mutations on the accumulation of beta amyloid involved the presenilin knockout mouse. And again, these mice were deficient in presenilin. These are early days in the late 1990s, before the modern molecular techniques were available, and these didn't last very long. They died quickly after birth, which meant that for looking at the effects with aging, these mice weren't very valuable to us. However, they did exhibit, before they died, massive neuronal loss, as well as different hemorrhages that were found in different areas of the brain. So again, not a perfect model. Again, multiple gene transgenic mice where you combine presenilins, APP overexpression, and you combine all of these together. When they start doing this, they actually start to get slightly better results. When they looked at APP plus presenilin knockouts, and when they started looking at the overall expression, exactly as you'd expect, more APP. The toxic fragments of it. And again, both young and old animals in this particular transgenic with the APP presenilin have deficits in what's known as this spontaneous alternation task in the Y maze. And I'll get to that in a moment. If it's something new to you, it's okay. I'll get to that in a moment. And again, fewer alternations between the arms in the Y maze. The better, or the higher the number of alternations where they stay in one arm before they switch to another is actually better. It's a better form of memory. So the lower the number is, the worse your memory is, and I'll get to that in a few moments. Again, there are lots of issues that actually exist with this particular mouse. It's still used, by the way, in the literature. You'll see it from time to time. And again, there are some good parts to it in the sense that this has a strong positive correlation between the more of this toxic fragment that you generate over time with age, the more cognitive decline that you'll see in these transgenic models. The last one I'll get to before I get into the APP 23 mouse is the CRND8 mouse. And this is the last and possibly the most widely used transgenic mouse. The CRND was actually developed here in Toronto. And the CRND8 mouse is actually derived from a Swedish mutation in APP, where the valine is substituted by phenylalanine at position 717. And as a result, they start forming plaques in the hippocampus and cortex around 9 weeks of age, a little earlier than some of the models that I'll show you, or the next model that I'll show you. Again, these mice, the transgenic CRND8 mice, differ from the ones that I just showed you in the sense that they have dense core deposits that I'll give you a picture of a little bit later on, and dystrophic neurites that look like they're damaged without any changes in the hippocampal volume. So again, not a perfect model overall, but it's a better model in some ways. In the Morris water maze, or the MWM, and we've talked about the spatial Morris water maze, hippocampal dependent, the CRND8 mice perform worse than their control counterparts after the plaques have developed. Behaviorally, they respond in the way that we think they should also respond. Again, it seems to fit in with this amyloid cascade hypothesis in the sense that they perform worse depending on the stage that you look at them, before plaques, after plaques have formed, etc. So the last one that I'll talk to you about, and hopefully you've remembered some of the models or some of the details of the material and methods in this particular paper. The APP23 transgenic mouse also overexpressed the human APP. There is a Swedish double mutation. So it's not the single valine to phenylalanine switch. There is a double mutation that goes on here. These mice start to develop amyloid plaque pathology at about 6 months of age. They will later have vascular amyloid deposits, so they'll also have not only neuronal dementia, they'll also have vascular dementia as well. Plaques are found to be the compact, dense core type, and these increase exponentially. The other thing to make note of, female mice actually show a more rapid A beta plaque deposition than do males. So again, one of the reasons why you separate out males and females is the timing, as well as the acceleration of the plaque deposition. Males tend to be on a slower time course using this APP23. Again, associated with this, we have all the things that we want: synaptic degeneration, tau hyperphosphorylation. In many ways, this is a better model system than all of the ones that I've shown you before. So right now, most people either use the APP23 or a variation of the CRND8 mice. Okay, so I gave you a really broad overview of the different types of transgenic mice. You should be familiar with some of the deficits and the advantages of using the 5 different models, and understand that this APP23 and the CRND8 mice are important right now in terms of research. And this is why, instead of using any of the other ones, they stuck with using this APP23 mice. And again, one of the things that they looked at was these 10 male mice that had the plaque-forming APP23 transgenic mice. Again, at a certain time frame within the 12 to 13 months that I talked to you about before, they already have a substantial plaque burden. It's not overwhelming, but they've already started to develop these plaques. Again, different between male as well as females. We've already talked about the weeks, the sham, and over this 4-week period where they do this testing, it's important to realize that these are males that have a plaque burden already, and can we reverse some of that? One of the behavioral tests that they do is what's known as this Y-maze alternation. So again, the Y that you're thinking of, the 2 arms are actually fairly short at the top, and then there's a longer Y arm, the longer vertical line. In fact, they're all identical in length. It's like having a triangle almost, where all of the arms are identical in size, and they want to be able to explore all of the different arms in this new environment. And one of the things that you can look for is when you place this animal in this Y-shaped maze in the middle, and then you allow them to get habituated to the maze, then you'll go back and record the number of arms that get entered, as well as the sequence of entries into these individual arms. You label them A, B, and C, for example. You record them, and then you calculate what's known as an alternation rate, the degree of arm entries without repetition. In other words, the animal has to remember, I was already in arm A, maybe I want to go and look at arm B, and then maybe I want to go and look at arm C. And the higher the rate, the alternation rate is, the better they remember that they've already been in one of those arms. This higher alternation rate is actually indicative of sustained cognition, as this animal has to remember which arm it has already entered. And so if you go back and look, this is a non-transgenic animal, so there is no burden of plaques or anything else. This is what you would expect at baseline if the animal remembers perfectly, and their alternation rate is sort of what you should be looking at. Remember, higher Y-maze alternation rates mean better cognition, better memory. The APP23 that has already got a plaque burden, that already looks like it has pseudo-Alzheimer's, has a lower alternation rate, meaning that its memory is affected. Its cognition is already in decline. If you go back in and look at the APP23 after 4 weeks of the scanning ultrasound, they actually go back to baseline levels. They improve behaviorally on this particular test. So in other words, their cognitive decline that they normally would be expected to have has actually been reversed. So why is it behaviorally that they are looking like they are pseudo-normal mice? So one of the things that the authors wanted to do is, has this actually gone and affected what's known as the plaque load? Have we actually changed the number of plaques that these mice normally develop? And is this the reason why they are actually performing better? And one of the things that they did was a standard histological technique known as the Campbell-Switzer staining. What other silver staining methods do you know of? Should know at least two as neurobiologists, at least two. So all of you know the Golgi silver impregnation, right? You guys all learned the basics of silver impregnation. You incubate in potassium dichromate, and then you put in silver nitrate, and you get the infiltration into neurons. The other one, what's the other standard histological or histopathological silver staining technique? Anyone from LMP here? No one from LMP here? Yes.
I don't know if it's silver, but
yep.
In HMB300 there was a staining substance called gadolinium.
Yep. It's a slightly different stain. It's a variation on the silver staining, but the one that you'll see most often in pathology is either going to be the Campbell-Switzer or what's known as the Bielkowski type of stain. And again, you don't have to worry about that. I was just more curious than anything else. So one of the things that they looked at was, is there a reduction? And in fact, after scanning ultrasound, they were able to reduce it in the cortex by 56%. And again, overall, they were able to reduce it by half, which is why they probably performed a lot better compared to sham animals. And again, if you go back and you took a look at the overall silver staining, this is what the normal transgenic sham mice would be. They get the microbubbles, they get the dye, they get nothing else. There's no ultrasound applied. And you can see the plaque load is actually fairly intense up in the cortex, but after ultrasound, it actually is reduced substantially. And these are what those dense core silver stains look like, as opposed to the diffuse type that you look at later on. And again, the plaque burden has decreased substantially by half just by using microbubble injection and by having scanning ultrasound. And again, it's remarkable that they're able to do it. It's a non-invasive technique. No antibodies are applied to this. All they're doing is opening up the blood-brain barrier by using microbubbles, and that's it. That's all that they're doing. So is it actually a reduction in A beta plaques? Maybe one of the things that happens is you get less of those silver deposits, but maybe it has nothing to do with the A beta. And one of the things that you can do is go in and use a really old stain known as thioflavin S, and it will just be applied to your sections, and it will bind to amyloid. And it binds to amyloid really well. And it fluoresces. It's a chemical that fluoresces normally, and you can see that in thioflavin S staining in sham versus the scanning ultrasound, there's also a reduction in beta amyloid. It's not just plaques, it's beta amyloid containing plaques that the ultrasound is causing to be removed. They further confirmed this by using an antibody-mediated technique. The A beta is recognized by an antibody known as 4G8. It is specific only for A beta containing plaques. And you can see exactly the same thing as thioflavin S, and was suggested by the earlier silver staining that in this particular stain as well, there's also reduction in the A beta plaque load as well. So it was very convincing that plaques are gone, A beta is gone from the brain. So again, using some more sophisticated protein chemistry techniques, they start Western blotting with different antibodies against the A beta. Do you guys all take biochemistry? Yes? So what does this mean? I'm always curious. You don't have to know any biochemistry per se. If you do, that's great. They actually take two different fractions. So they take one which is known as an extracellular fraction, and then they take what's known as a Triton soluble fraction. What's the difference between the two? Like if you're reading through this paper and you suddenly encountered, they did like fractionation, there's a Triton soluble, and then there's this extracellular soluble fraction that they looked at. What does it mean? Yes? It can be used to permeabilize cells, but it's also used as a biochemical way to break up membranes, and it's there to break up insoluble insoluble fragments that are found inside cells. So in other words, what they did, and this looks like it's exactly the same, it's not exactly the same. This is the extracellular, outside the cell type of amyloid beta, and this is all the stuff that's found inside, the Triton soluble material that's found inside the cell. Regardless of which, you don't have to memorize this slide. This Western blot, using a number of different antibodies, shows that either inside the cell or outside the cell, when you take the brain after sonication, one of the things that happens is that there is a reduction either inside or outside the cell in different size fragments of beta amyloid. So you can have high molecular weight beta amyloid, the full fibrils in other words, okay? Or you can take the smaller fibrils or the smaller integral parts of the larger fibrils, and all of them tend to be reduced both inside and outside the cells. If you go back and you look at it as a whole, all of the beta amyloid in total, whether it's inside or outside of the cell, or in the soluble or insoluble fraction, that there is an overall reduction here as well. And again, you don't have to worry about all of the details. Some of that's on the next slide, but these are just different size species. Again, think of the oligomers that eventually form the fibrils that eventually form the insoluble plaques, similar to what you heard from Dr. Kalia on last Thursday as well. It's exactly the same process. You start with small soluble fragments that form larger insoluble fragments that eventually form these plaques that might be involved in cell death. So all of these different isomers, whether it's the high molecular weight species or the 56 oligomeric A beta or the trimeric A beta, which is just the toxic APP fraction, all of them, regardless of where you're looking, after scanning ultrasound, every last species is reduced. Pretty remarkable. In biology, in order to get published in a high-impact journal like Science Translational Medicine, you also have to have a mechanism, or at least a putative mechanism that might account for this. They don't really know this 100%, but one of the things that they speculate on is that scanning ultrasound actually increases the ability of the microglia to cause the clearance of these amyloid beta fibrils, whatever the heck these fibrils are, larger or smaller, okay? Important point, it's not an increase in microglia. In fact, they go to great pains to show that after you break down the blood-brain barrier, the microglial numbers stay exactly the same in the brain. It's not an increase in microglia, they just become more efficient. And one of the things that they saw, what do you observe in this sort of camera lucida type of drawing down here? So these are all microglia that have been stained using an antibody, and then these are all of the sort of different outlines of the cells that you can actually trace using a computer program. Do you notice anything at all between the transgenic, non-transgenic wild-type mice, no APP at all, the APP23 sham, as well as the ones after ultrasound? Yes?
They're more dispersed on the SUS.
So more dispersed. Okay, maybe. So each one of these represents one microglia. So this is right on top of it, there's the microglial cell body, and then the little processes going off, the little arms going off. Do you notice anything different? Yes. So the more processes after SUS? Fewer processes. They become shorter. In fact, that's one of the things that they see is that the processes actually become smaller after SUS. So you stain the microglia with this antibody known as IBA1, and after IBA1 staining, you do a computer outline, and one of the things that they see is down here on the bottom that the processes actually become shorter. The process lengths become shorter, the processes overall become shorter in these activated microglia following SUS. And they think that one of the things that happens is by shortening the processes, they're a lot more mobile. So imagine like you have these long arms and you're trying to get through a turnstile, you might get stuck. But if you have these shorter arms, you might be able to move a lot better. You're more motile. And in addition to that, Again, we won't worry about the details up here at the top in too much detail, but you can actually go in and show that following SUS that you can see more internalization into the microglia of the beta amyloid. So they're not only more efficient, they are also internalizing much more of the beta amyloid after the scanning electron microscopy. So then why does it do that? Why does simply opening up the blood-brain barrier do all of this? It was a really big surprise to these individuals. Using a different source, using a cell line, this is not found endogenously in your brain. Just like you have neuronal cell lines, you also have microglial cell lines, and this microglial cell line is known as BV2. And when you go in and take a look at BV2 and you look to see if they will internalize beta amyloid, if you want them to really internalize beta amyloid, you add in albumin. Guess what gets into the brain after you open up the blood-brain barrier? Albumin. And so just by doing this, by adding albumin, you increase the activity of these microglial cells. And this is putatively one of the mechanisms by which this all could work. The microglia get activated, they endocytose better with the addition of a substance that's found in your body normally, this albumin. And again, remarkable if it turns out to be true. It still has to be replicated. It has to be replicated by other labs. But one of the things that was very clear on this is that SUS engages microglia, does not increase their numbers, but promotes the internalization of A beta into microglial lysosomes to allow them to be cleared. It restores the function in different behavioral tests like the Y maze, as well as other forms of memory that I didn't get into here. And most importantly, no inflammatory changes whatsoever were observed in these SUS treated mice, suggesting that if this is all true, and again, it has to be replicated and it has to be repeated, and that's why you publish in a high impact journal like Science, that if it turns out to be true and all of this works and the next clinical trials on humans work, that this is actually a viable therapeutic strategy for human subjects that have Alzheimer's disease or any neurodegenerative process by a non-invasive mechanism. And again, it's pretty remarkable that the pace of change in neuroscience occurs so incredibly rapidly. So we have a guest speaker on Thursday, and again, I went through a lot of different slides today. Think about them. I'll be here on Thursday to answer any questions, and if you have any others, please feel free to let me know. No, no, they have not.
Okay. So would that be like a future test, or would
Possibly.